Today we're here with Dr. Christie Mayo. She is an assistant professor and virology lead for the Veterinary Diagnostic Laboratories. So thanks for being with us today, Christie.
Thank you. It's a pleasure to have you.
Tell us a little bit about what you guys do here.
So, virology, this is the study of viruses, and a few key examples of those happen to be things you might have heard about in the public. Avian influenza, been quite popular recently. The other one might be BVD for the local practitioner. And so what we do is try to get samples in here, and then the everyday turnaround is usually a day within maybe to a week, but we try to process those samples and identify answers for the veterinarian that submits them. So the testing we do when we're trying to find what might be causing an abortion in a cattle herd or what might be causing respiratory distress is we'll do a series of either serology, which is analyzing serum, or maybe tissue samples, and we'll try to understand, okay, what virus might be causing this problem, and keeping in mind it may not be a virus at all, so sometimes we can get negative results. One of the biggest things that I enjoy about this job and this position is I get the fun exercise every day of being a puzzle solver, just basically getting in there, trying to answer the questions, and I get to use a variety of techniques. And so virology started a long time ago, you can imagine, and so we still use some of those fundamental techniques, sometimes kind of archaic, but things like virus isolation, utilizing real living things, eggs, cells, to grow these little buggers, and so that's a lot of fun for me. The next phase is where I've come in, trying to adopt new technology. So next-gen sequencing is quite popular, especially in the cutting-edge literature.
Right.
We're trying to use CRISPR-Cas9 pathways to do genetic, kind of alter the genetic structure and get host sequence reads out of that material. And what I like about this particular area is that being in the center, kind of the United States, you get samples from all over.
The massive changes that I'm sure you guys are seeing in diagnostics, it has to be just amazing to be working in this environment.
Oh, it's fabulous, especially when you're given the opportunity to seek that answer. And I just went to an innovation summit this past weekend, and the way technology is changing, it's an exponential fold. And so for us to be able to keep up with that, with novel toys, much like NextSeq and MySeq platforms, that's the nice thing. The other thing that we have here that maybe other diagnostic labs might not is this amazing resource of students.
Well, kind of coming back to what you guys are doing here, how many samples are you guys processing for virology on a daily or weekly basis?
Yeah, so it can range. There's a variety of different things that we do, but usually our statistics are about 30,000 tests within a year. And so it's a big variety as well because it's small to large animal. And so we do everything with regulatory testing. We do classic rabies testing for dogs and cats, even wildlife. When we look at some of these diagnostics, we really value our equine infectious anemia cases. Those are regulatory for transport, and so we do that quite often. Here in the spring is a really hot spot. I think we had 830 just last month. Being able to process, the technicians take the great burden, but also exercise and pride in delivering every single diagnostic test and result back to the producer, and that's what warms my heart. We often create a kind of what I hope to be a lifetime of client communication. So being able to pick up the phone, call me personally and say, hey, I just didn't get this result at all. And that's where I get a chance to say, it's sometimes gray, there's not black and white. And we go to the next level, and so maybe we can ask, maybe there's another test. And so that's the fun part. And sometimes they can enroll in some of my research. So maybe we don't have the answer, but I can move them over into the other side.
Well, why don't we take a few minutes and just take a look at some of the actual tests that you guys are performing.
Sounds great, let's do it. So we have Monica here doing what's a classic virology technique called serum neutralization. And so what we do this for is to actually get what is called a titer. So we want to know how much antibody is in the body of an animal. We're sitting here at the fluorescent microscope. This is what we usually use to do fluorescent antibody testing and evaluation. So what's happened up until now, if we have a classic case of rabies, we'll take a bit of the brainstem from down on the necropsy floor. So that comes in. We rush it up to here as fast as possible, especially if there's been human exposure. And we take great consideration for doing the staining. It takes about 3 hours. And then we finally evaluate it under the scope. I'd have to turn the lights out and the fluorescence on in order for you to see it. But it's really neat because it glows like apple green. And so if there's a positive, that's not so much fun. But usually in our wildlife species, we can test quite a few positive rabies in a year. And that will be reported to our public health agency.